Hello everyone. Today's topic is HMP shunt. It is also called as hexomonophosphate shunt. This pathway is occurred in cytosol. As we know that glycolysis is occurred in cytosol. That's why HMP shunt is an alternative pathway for glycolysis and TCA cycle where we can get energy molecules ATPs. But in HMP shunt is not producing any ATP molecules, but it is very much important pathway. It produces NADPH and ribose 5-phosphate, which are very much helpful for formation of few important biomolecules such as fatty acids, nucleic acids, coenzymes, etc. So choosing of these pathways is very much important. So it is depending upon current needs of the cell. For example, if a cell is rapidly growing, it requires more amount of biomolecules such as fatty acids, nucleic acids, cofactors, etc. So in that case, so cell chooses HMP pathway. If a normal cell is there, which carries many metabolic reactions, so it requires more ATP molecules, more energy for carry out of these metabolic reactions. In that case, so cell chooses glycolysis and citric acid pathways. So it is depending upon current needs of the cell, right? So coming to the HMP shunt pathway, today we are going to discuss about HMP shunt pathway. So this is HMP shunt pathway, which is very complex pathway. It is not a top to down pathway like glycolysis. So here many intermediates are involved. So here this HMP shunt pathway is divided into two important phases. The first one is oxidative phase, so in which oxidation is carried out. And the second one is non-oxidative phase, where many intermediates are interconverted and forming a new intermediates. Right? Coming to the oxidative phase, which is also called as non-reversible phase. Here we have three reactions. So all reactions are non-reversible. Coming to the first reaction, glucose 6-phosphate is converted into 6-phosphogluconolactone. Here, a linear chain glucose 6-phosphate is converting into a lactone ring. So this reaction is catalyzed by glucose 6-phosphate dehydrogenase. This is an oxidation reaction. Here, glucose 6-phosphate is oxidized. So NADP plus is involved in this reaction, which is a coenzyme. Here NADP is reduced. It is taking two hydrogens from glucose 6-phosphate and converting into NADPH. So this is a very much important reaction. It is a key reaction, and glucose 6-phosphate dehydrogenase is the key enzyme, which is also called as regulatory enzyme or rate limiting enzyme. So by this process, NADPH will be produced, which is very much essential for formation of fatty acids, cholesterols, and sterols. Here, one important point is there that many students are getting confused that NADPH will be producing ATP molecules. No, so this is not. NADPH will not be entering into electron transport chain. It will not produce any ATPs. NADH will be produced 3 ATPs, but NADPH will not produce any ATPs. Coming to the second reaction, here the 6-phosphogluconolactone, which is a ring structure, is converted into 6-phosphogluconate. Here the ring structure will be broken down, and this is hydrolysis reaction. Here water molecule is added, and also this reaction is spontaneously occurred. So this reaction is catalyzed by gluconolactone hydrolase. In the third reaction, 6-phosphogluconate, which consists of carboxyl group, is converting into a 5-carbon ribulose 5-phosphate. A 6-carbon compound is converting into 5-carbon compound by releasing of one carbon dioxide molecule. So this is called oxidative decarboxylation. Here 6-phosphogluconate is oxidatively decarboxylated and forming 5-carbon compound, that is ribulose 5-phosphate. Here also one more NADP is involved and forming NADPH. So this reaction is catalyzed by dehydrogenase enzyme, that is phosphogluconate dehydrogenase, in the presence of magnesium ions. So now we are forming ribulose 5-phosphate, which is a pentose sugar. So these three reactions are coming under oxidative phase. Here, by this oxidative phase, finally we can get two molecules of NADPH and one molecule of carbon dioxide. Coming to the second phase, that is non-oxidative phase, so which is also called as reverse phase. So here all reactions are reversible reactions. So this is the first product in reverse phase, non-oxidative phase. Here ribulose 5-phosphate, which is having 5 carbons, which is converting into xylulose 5-phosphate, which is also having 5 carbon. So that means these two compounds are epimers to each other. Here ribulose 5-phosphate gets epimerized and forming xylulose 5-phosphate. So both intermediates are having the same number of carbons and same functional group is present, in which uh, ribulose is having keto function group and xylulose is having also keto function group. So where the change is occurred? Third carbon position, the configuration is altered. So that's why these two compounds are get epimerized. So this reaction is catalyzed by ribulose 5-phosphate epimerase. In the next reaction, ribulose 5-phosphate is also converting into ribose 5-phosphate. So ribose 5-phosphate is having aldehyde group and ribulose 5-phosphate is having keto group, meaning ribulose 5-phosphate gets keto isomerized and forming ribose 5-phosphate. Aldehyde group, sorry, keto group is converting into aldehyde group. Now we formed two intermediates. One is xylulose 5-phosphate and the second one is ribose 5-phosphate. So both intermediates are having 5 carbons only. Now 2 carbons will be transferred from xylulose 5-phosphate to ribose 5-phosphate in the presence of trans-ketolase enzyme, which is coupled with the TPP, that is thymine pyrophosphate, which is a coenzyme. So when xylulose 5-phosphate transfers 2 carbons from this molecule, so automatically this xylulose 5-phosphate gets converted to 3 carbon compound, that is glyceraldehyde 3-phosphate, and ribose 5-phosphate, which is having 5 carbon, which accepts 2 carbons, so then it is converting into 7 carbon compound, that is pseudoheptulose 7-phosphate. So this reaction is catalyzed by trans-ketolase. This is very much important reaction. So this reaction is carried out in the presence of TPP. If the cell is lacking of thymine pyrophosphate, we cannot produce pseudoheptulose and glyceraldehyde 3-phosphate. In the next reaction, here these two intermediates are combined and rearranged by trans-aldolase enzyme. Here 3 carbons will be transferred from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate and forming erythrose 4-phosphate, which is 4 carbon compound, and fructose 6-phosphate, which is 6 carbon compound. Now in the next reaction, xylulose 5-phosphate, which has 5 carbons, so which is having keto group, transfers 2 carbons to erythrose 4-phosphate and forming fructose 6-phosphate, which is having 6 carbon compound. So erythrose is accepting 2 carbons and converting into fructose 6-phosphate. And xylulose 5-phosphate is given 2 carbons and converting into 3 carbon compound, that is glyceraldehyde 3-phosphate. Now we got two important final products which are produced from HMP shunt, and these two final products will be intermingled with the glycolysis process. Why? Because these two compounds are also present in glycolysis pathway, and this important precursor is also present in glycolysis pathway. So this is completely a complex pathway. So here many reactions are intermingled, and it is leading to many other processes, metabolic processes. So this is about the HMP pathway. This is very much important for the cell for biosynthesizing fatty acids, cholesterols, etc. Here ribophosphate. So ribose 5-phosphate is very essential for synthesizing nucleic acids such as DNA, RNA, etc. And also it is synthesizing nucleotides, ATP, NAD, FAD, etc. So here in HMP shunt pathway, so NADPH and ribose 5-phosphate, so these are the main important products will be produced. And erythrose 4-phosphate is also one of the important intermediate which will be useful for synthesizing aromatic amino acids. Here in non-oxidative phase, all reactions are reversible nature. So sometimes erythrose 4-phosphate gets converted into xylulose and ribose 5-phosphate. These are all reactions are reversible, but whereas in case of oxidative phase, here reactions are irreversible. So this is about HMP shunt pathway. Thank you all. If you have any doubts, please ask me in the comment section, and don't forget to subscribe my channel. Thank you.